Sorry. Did I share my presentation now? Am I able to share my presentation?
Dr. Geet, I also missed our presentation yesterday. So
Am I able to share the
if there's an
presentation?
opportunity, I can also present right now.
Hello? Hello?
Yes, please go ahead. Dr. Bilal, please go ahead.
Am I able to share the presentation? Are you able to see the presentation?
If, Dr. Bilal, you should present. Yesterday, I had mentioned that you should.
I mean, am I able to share my presentation? I'm trying to share the presentation. Is it
PDF file, not the PPT file.
not the PPT file? That is what.
You're sharing a PDF file.
Yes. Unsupported document.
चलिए डॉक्टर साहब आप यही प्रेजेंट कीजिए. We are losing time. Please present the same one.
Okay. Okay. A very good afternoon to all you all, and respected Director HRD University of Kashmir and Coordinator Dr. Gir Saab and my fellow participants. Today I am going to present something about remdesivir, a potential antiviral drug for COVID-19 treatment. As of now, everybody must be knowing about the coronavirus and the devastation it has caused across the globe. So I will briefly discuss about some things and the development of remdesivir as a potential therapeutic drug against the coronavirus. So before I will start, I will just introduce something about coronaviruses. So coronaviruses are a family of enveloped viruses characterized by a club-like spike that project from their surfaces. The word corona in Latin means hollow or crown. These are single-stranded RNA genomes that infect animals and other animal species and the humans. They belong to the family of Coronaviridae and are further subdivided into four classes: alpha, beta, gamma, and delta classes. So, among globally identified coronaviruses, only four types of coronaviruses were known to cause only mild infections in humans. They are including 229E, NL63, and OC43, and HQU1. So till date, none of the alpha and beta forms have been identified to cause any infection. However, the notion has changed with the discovery of two new strains of coronaviruses that caused outbreaks of SARS pandemic in 2003 in China and MERS pandemic in 2012 in Saudi Arabia, with the death rates greater than 10% and 35% respectively. These strains of coronaviruses are believed to have emerged from the native bat populations and transmitted to the humans. In recently in December 2019, a novel strain of coronavirus, SARS coronavirus 2, was reported to have emerged from Wuhan, China's Hubei province, belonging to the same family of viruses that caused SARS and MERS, as well as other four human coronaviruses. And evidences also suggest that these coronaviruses also reported from, emerged from bats or fish market of China. So the disease caused by this coronavirus strain 2 is termed as COVID-19 by WHO. The acronym derived from the coronavirus uh, disease, the coronavirus disease in 2019, the year in which this pandemic emerged. Since its outbreak, it has caused huge scale of devastation throughout the world. Millions of the people lost their lives, and billions of the people got infected by this virus. And on March 11, 2020, WHO declared COVID-19 as a global pandemic. Initially, it was also declared as global health emergency in June 2020. So, further, And further research is needed. That is needed to to work on this coronaviruses and other virus strains. So the further research has revealed that SARS-CoV-2 strain 2 shares almost 80% of RNA sequence homology with the SARS coronavirus 1, and the enzyme RNA dependent RNA polymerase is present in both viruses and shares almost more than 95% of the sequence similarity between these pandemics. Thus, it appears that drugs that are effective against the treatment of SARS and MERS may also have a potential, or they can be also harnessed for the development of drugs against this COVID-19 disease. So researchers worldwide are searching for effective therapy or treatment of this disease. Since all of us know that development of a drug or development of a vaccine against a particular viral infection, it takes almost a decade. But however, it takes almost a decade. However, all of us have heard about the development of vaccine by Professor Parvez Saab in his inaugural lecture. So it takes almost 10 years. But in this case, however, only 1 year it has been reduced from decade to 1 year. So under these circumstances, when the drug development takes decades and the vaccine development takes decades, so repurposing of the existing drug could act as an alternative to combat the disease effectively. So in this regard, several classes of pre-existing drugs have been repurposed for the treatment and are in various stages of clinical trials to address the current SARS-CoV-19 2 pandemic. So one example of drug repurposing is the chloroquine and the hydroxychloroquine. Both these drugs are derivatives of quine as well as a well-known anti-malarial drug. However, these two derivatives of quine have also been developed against malaria. Although nowadays both these derivatives have lost their antimalarial efficacy because of the development of resistant Plasmodium falciparum. So, after the outbreak of SARS and MERS pandemic, these molecules were found to have highly effective against the viruses. So, keeping this in view, these two molecules were tested for SARS COVID-19 2 coronavirus 2 strain and were found to be highly effective in vitro and in vivo studies. So keeping the potential of this in view, on March 2020, FDA issued an emergency authorization for the use of hydroxychloroquine and hydroxychloroquine for the patients hospitalized with COVID-19. This move was supported by the Trump-led U.S. administration in White House, despite having scanty evidences. In India, also, India has also taken an interesting position on the role of hydroxychloroquine and chloroquine for COVID-19. The National Task Force for COVID-19, constituted by Indian Council of Medical Research on 22 March 2020, also recommended the use of hydroxychloroquine for prophylaxis of SARS-CoV-19 infection for the healthcare workers and household contacts of COVID-19 patients. So there have been a sharp difference of opinions as to the role of these two drugs in the prevention and treatment of SARS-CoV-19 2 infection. One On one extreme, these drugs have been rooted as the biggest game changers in the history of medicine, while on the other hand, these drugs have been trolled as useless and dangerous. The results of preliminary large-scale randomized controlled trials have failed to show any survival benefit of such drug therapy in COVID-19. Based on these results, as of today, most experts are against the use of this hydroxychloroquine in the treatment. Therefore, in June 2020, FDA withdrew its recommendation of use of hydroxychloroquine for COVID-19 patients. The other drugs that are already in pipeline that are already repurposed for the treatment of COVID-19, they include some known antiviral drugs, for example, ritonavir and lopinavir, ribavirin, and favipiravir. These different types of drugs that are in clinical trials, they belong to either they are proteases or they affect the RNA polymerase that is used for the synthesis of RNA viral RNAs. And the important that I am going to discuss is the remdesivir. So remdesivir is also an existing drug. It's a phosphoramidate product of ribose triazine C nucleoside. The name, the technical name GS441524. It was developed for the first time by Gilead Sciences in collaboration with the CDC and USAM RIID, with a research program in HVC, hepatitis C virus, and RSV, respiratory syncytial virus. It was found to show broad spectrum antiviral properties as it is an RNA polymerase inhibitor against various types of viruses. Then, with the outbreak of SARS and MERS, Gilead Sciences, in collaboration with both the academic institutions, expanded the antiviral profile of this molecule against these viruses. So after the Ebola outbreak, Gilead Sciences provided remdesivir for the treatment of small number of patients with Ebola infection under a compassionate protocol. All these studies confirmed that remdesivir was active against these coronaviruses in vitro laboratory tests and in vivo preclinical animal models. Even though it had positive preclinical results, remdesivir could not be advanced into clinical development for SARS and MERS due to lack of adequate number of potential study participants. The number of MERS infections was limited with almost exclusive location in the Kingdom of Saudi Arabia, and there was no SARS infection in Jan 2020. After the outbreak of COVID-19, Gilead Sciences moved quickly to initiate the studies to evaluate the remdesivir for its safety and efficacy in the treatment of COVID-19. In preclinical studies, remdesivir was found to show very good results, or we can say they have shown some promising results. So the molecule, because of its very good potency in the in vitro studies, was repurposed for COVID-19 treatment. So on May 1, 2020, the United States Food and Drug Administration granted emergency use authorization for remdesivir to treat COVID-19 patients. Now, how it works basically, this remdesivir. So the parent nucleotide has less permeability on the cell membrane, so its product form was synthesized that easily diffuses into the cell, where it is first converted to alanine, metabolite by the action of esterases, followed by the action of imidases, leading to the formation of nucleotide monophosphate, which undergoes further phosphorylation to form the nucleotide triphosphates. And these triphosphates mimics the activity of basically actual ATP and act as a substrate for many RNA-dependent RNA polymerase complexes, and has been reported to inhibit the viral synthesis by specific mechanism of delayed termination, as for few nucleotides get added and later on the drug gets dissociated from the RNA-dependent RNA polymerase. These are some evidence of the in vitro antiviral data of remdesivir, which has been shown to have potential effect on various virus strains across the globe. Similarly, the summary of broad antiviral and in vitro antiviral activity of remdesivir against human viral pathogens has been studied also, and it has also shown a very promising effects or efficacy. The evidences, or there are some evidence of animal studies of remdesivir on coronaviruses. They include the in vivo study using the Ebola-infected rhesus monkey. Another study was conducted on the mouse model of MERS COVID-19, and next study was conducted on the mouse model of SARS-CoV-1. And recent study was conducted on the rhesus macaque model of SARS-CoV-2 coronavirus 2 strain, where the authors have demonstrated that animals treated with remdesivir did not show any signs of respiratory disease and had reduced the pulmonary infiltrates on the radiographers compared to the control animals. Some of its human cases include, the human studies include, in all the cases, it was given intravenously with a dosage of 200 mg on day 1, followed by 100 mg for up to 9 more days. The first COVID-19 patient reported to receive intravenous remdesivir was 35-year-old man in US who had traveled from China. He was hospitalized and his condition was very deteriorated. On the day 7th of the hospitalization, he was administered with remdesivir, and his condition was reported to improve on day 8 with no adverse events related to remdesivir. Another case study was also conducted on 42-year-old man who was on mechanical ventilation. On 13th day, he was administered remdesivir, and his condition was also reported to improve after 24 hours and was successfully able to transit to room air. Another report is the first 12 COVID-19 patient is diagnosed in US on March 12, 2020. Three of the 12 patients is treated with remdesivir under compassionate use for duration of 4 to 10 days, exhibited improved respiratory symptoms. These are ongoing clinical trials, and number of clinical trials are ongoing to determine the safety and efficacy of remdesivir. Preliminary results of the various ongoing clinical trials have shown that remdesivir administration led to faster clinical improvement in severe COVID-19 patients compared to placebo or standard care. However, results from large-scale clinical trials are important to confirm the efficacy of this remdesivir in treating patients with COVID-19. Thank you very much.
Thank you very much, Dr. Bila, for this presentation on remdesivir. So we are, very delighted to have with us Professor Ziaur Rahman. We wholeheartedly welcome him to this particular session of the day. And I also welcome Professor Mushtaq Ahmad Darzi, the Director in charge Human Resource Development, University of Kashmir. So to begin, we will just begin the proceedings of the first session of the day. That will be delivered by Professor Sayyid Ziaur Rahman. Uh, Let me have the privilege and honor of introducing Professor Sayyid Ziaur Rahman. Professor Rahman is working in the Department of Pharmacology, Jawaharlal Nehru Medical College, Aligarh Muslim University, Aligarh. He has been working in the field of pharmacology for more than 2 decades with vast research experience in basic and clinical pharmacology at both undergraduate and postgraduate levels. As a scholar, Professor Sayyid Ziaur Rahman has to his credit 7 published books, 17 chapters, and more than 200 research papers, case reports, and communication in both national and international journals. Professor Rahman has attended 63 workshops, training programs, and presented papers during 70 international conferences and 85 national conferences. He himself has organized 3 international conferences, CMEs, and 20 national conferences, symposia, and has handled 7 academic projects. Professor Rahman has earned his graduation, that is MBBS, in 1995 and MD in 2000 from JNMC Aligarh Muslim University, and has earned his doctorate from WSU, Western School of Medicine, Western Sydney University, Australia, and has contributed in medical science in the area of alternatives to animal experimentation, traditional medicine, and pharmacovigilance, and has won many laurels across India and even abroad. With this very brief introduction, we welcome Professor Sayyid Ziaur Rahman and wholeheartedly thank him for accepting our invitation. Well, ladies and gentlemen, as all of you know, today we are witnessing very severe, heavy snowfall, and there is also a power breakdown all across the valley. So, in spite of that, this science festival continues, and we are going to have hectic deliberations on the important issue of antibiotic resistance. That will be the topic which Professor Sayyid Ziaur Rahman will be discussing today, antimicrobial resistance. But before I invite Professor Sayyid Ziaur Rahman to begin with his talk, may I?
Yes. Yeah.
Yes,
Okay.
may I-
Okay. Dr. Geet, can you hear me? It's my
yes,
voice is audible. Okay, fine. That's good. So
just
thank
a
you.
minute.
Okay.
Just a minute. I would first invite our in-charge director, Professor Mushtaq Ahmad Darji Saab. I think he has joined us. Are you able to hear me, sir? Would you like to say a few words, Professor Mushtaq Saab?
Assalamualaikum.
Janab Gir Saab and Professor Saab, who is our. It is so nice to be with you, sir, this morning. Tremendous which are faced by our participants. But this time we are very much prepared, I suppose, that everybody has inverters or somebody may have power banks, and we can keep at least our mobiles on and will be benefited by the presentations of the speakers, which already has been scheduled by Gir Saab. And I hope that this festival will go on. We will not be this. We will not be. We will not stuck, and we will not be stopped by this festival because of the snow. And I hope that our participants will join us every day. Professor Saab, I welcome you, sir. It's very snowing this time in Kashmir,
Thank you,
and
Dr.
it's very pleasant, especially for you. But when we face difficulties, we feel that snow is not good, but at least this is actually the best thing which we are having in Kashmir. That's a snowfall. So thank you very much, sir. You have agreed to our request, and you accepted that you will have an interaction with our teachers. They are college teachers, excellence. They are.
Thank you, Dr. Gir Isaaq Saab and the coordinator, Professor Mushtaq Saab. I could hear this lecture, this introduction by Dr. Gir, but somehow I couldn't hear what Dr. Mushtaq said in the last because of some interruption and his voice was interrupting. So I don't know what happened. Anyway, so I hope my voice is clear and audible for all of you.
Yeah, Doctor, it was interrupted. Continue, sir. Continue, sir. It got
fine.
disrupted.
Thank you so
Yes,
much.
sir.
So
Your voice is clearly audible. Please continue and don't mind the disruptions from our side. You please continue.
Okay, fine. Dr. Giri, fine. That's good. So could you allow me to share my presentation?
Yeah, you are already allowed, sir. You can share directly. There are no restrictions.
So can you see the presentation now?
Yeah, it is about to come, but has not appeared as yet.
What about now? What about now? It's coming?
No, sir. Please
It's
try
visible?
to share it again.
Just a minute. All right, just let me see. Is it visible now?
Yes, it is visible now, sir.
Okay, that's great. That's
Please
great.
go
Okay, fine. Thank you so much. So at the outset, I would
Just
like
a
to
minute, sir.
thank all.
Dr. Bilal,
Yeah.
just a minute, sir. Sorry to interrupt. Dr. Bilal Ahmed Mir will moderate the session. So please take over, Dr. Bilal.
All right.
And
All right, we can begin our session today. And at the outset, I would like to thank the organizers who have given me a chance to share my views on antibiotic resistance and One Health concept, which we all know that there is a concern about the use of antibiotic and the resistance is becoming more and more alarming. So it's a good topic that we are going to discuss today. And the topic is the science of antibiotic resistance and One Health. So I hope Dr. Bilal may comment if I have some problem from here. So I'm going to discuss about all the relevant points related to the drug resistance as well as how to cope with the One Health concept. Okay, so at the outset, I would like to introduce that why this problem has been occurring. And you all know that antibiotic is a very useful armamentarium in the pharmacology, and it is used to prevent and treat the bacterial infections. But at the same time, we are witnessing antibiotic resistance with these bacteria in response to the use of these medicines that could be the overuse or not very effectively. So you all know that this resistance occurs in bacteria and not in humans or animals. So if the resistant bacteria may infect humans and animals, and you can imagine that at that time they will be hard to treat those infections which are happened because of the non because of the resistant bacteria comparative to the non-resistant bacteria. And because of this global problem, the world is urgently needed to change how we prescribe the medicines and use the medicines by the consumers, not only the antibiotics but also the other antimicrobials like antivirals, antifungal, and antiprotozoal drugs. So there is a need to change about the concept of the use of antibiotics and antimicrobials, and this also needs to change the behavior pattern of the whole population. And this behavioral change may include the spread of infection through vaccination, hand washing, practicing safer sex, and good food hygiene. So not only we restrict the use of antibiotics, but also we need to work on the behavioral change. And you know that antibiotics first came in around after the First World War, and the first drug was penicillin. And you can see that antibiotics usually remains in the world for a few years or to decades. Like for example, you can see that penicillin, which was discovered in 1943, but within 3 years, it became resistant to many bacteria. Similarly, you can see that sulfonamides came, but it took a decade to become resistant with this antibiotics. So there is resistance in almost all the antibiotics, which you can see from this list. So I'm not going to repeat the same thing. The resistant problem is there, and we need to work to find out the key facts of these antibiotic resistance. And we all know that this antimicrobial resistance, although antibiotic resistance is terminology is there, but more appropriate to say is antimicrobial resistance. So antimicrobial resistance is the biggest threat to now the global health, food security, and the development of the medicine. And this antimicrobial resistance is found everywhere, and in almost all the countries. It can affect anyone of any age group of the population, children's, geriatrics, or in the adult age group of the population. And this is rising dangerously to the high levels in almost all parts of the world. That is why WHO came up and working on the antimicrobial resistance policy. And this rising to dangerously high level is because of the ease and frequency with which the people travel all over the world, and they carry the resistant bacteria. So newer and newer resistant mechanism is also coming out and in these bacteria and spreading the whole world. And now there is a concern with the treatment of the common infections with the antibiotics, and these infections they become less effective as well as resistant to most of the common antibiotics. And it's very hard to treat even the common diseases like pneumonia, tuberculosis, gonorrhea, and salmonellosis. This antibiotic resistance occurs naturally or acquired or genetically. But apart from this occurrence of antibiotic resistance, there are other factors like misuse of antibiotics in both humans and animals without a prescription, and in some countries where the use of antibiotics is without the standard treatment guidelines. You know that WHO has also framed a model guidelines for the treatment, and all countries, all states, all cities, all hospitals need to have its own standard treatment guidelines, and according to the pattern of the usage of the sensitivity of antibiotics that should be available in all the hospitals. And as per these standard guidelines, then there is less chance of the overprescription or overuse of the antibiotics by the health workers and veterinarians. Next question comes why we are witnessing this antimicrobial resistance. Already misuse and overuse of antimicrobials in humans, animals, and plants. Uh, Poor medical prescribing practices and patient adherence to treatment. And you know that antibiotics kill the bacteria, but not the viral. So these antibiotics are not for viral, especially in colds and flu. So this is the poor medical prescribing, and we also know that because of the lack of standard treatment guidelines, and this leads to the incorrectly prescription in sometimes doctors overlook the medical conditions and they prescribe the antibiotics. And these antibiotics used as per farm animals and agriculture. Lack of the wash, which is the clean water, sanitation, and hygiene in humans and animals health care facilities, farms, and community setting. So wash is also noticed that there is a lack of sanitation and hygiene. And we also noticed that there is an inadequate infection prevention and control measures, uh, and this leads to the emergence and spread of the drug-resistant infection more. And the geriatric age group of the population, who is the renal or hepatic compromised patients, and also children who have immature immune system, they are having more problem with the increased use of the resistant bacteria. These days where we are witnessing COVID-19, and there is a high chance of misuse of antibiotics during COVID-19, as we just saw that these antibiotics are for bacterial and not in the viral disease. Um, we can see that these resistance is found in bacteria, viruses, and fungi. And if we see the bacteria, then we will see that the common antibiotics, like for example, you can see here, are used in the urinary tract infections, sepsis, and sexually transmitted infections and diarrhea. If we are having the resistant antibiotics, then resistant bacteria causing these infection, then there will be difficulty in treating these conditions. As we can see here that the rate of resistance of ciprofloxacin against E. coli varied from 8.4 to 92.9%. In some area, rate of resistance is 92.9% against. So if you can imagine that if E. coli is resistant to ciprofloxacin, then there will difficulty in treating the infection caused by the E. coli. Similarly, the rate of resistance with ciprofloxacin against Klebsiella pneumoniae to treat UTI is 4.1 to 79.4%. So this data is because of the WHO concerted efforts and now coming in the global scene. Similarly, the Klebsiella pneumonia, which is a common bacteria which may cause life-threatening infections if it is becomes resistant to most of the bacteria, and this resistant bacteria may cause problem in pneumonia and the blood infections and infections in newborn and the ICU patients. And if we are having resistance in Klebsiella pneumonia, then we are then have the sensitive drugs carbapenems. Uh, uh, Imagine if carbapenems also become resistant with Klebsiella pneumonia, then we don't have any other options. So carbapenem is the rescue drug, and in some countries, carbapenem antibiotics is also found resistant in the Klebsiella pneumonia. Similarly, in E. coli, uh, there is resistance because in E. coli, most of the fluoroquinolones are sensitive, but now they are becoming resistant. And in many parts of the world, these fluoroquinolones becoming ineffective. And then we are left if carbapenem is also resistant, then we are only having colistin. And imagine in some countries, colistin is found resistant. So, like Klebsiella pneumonia, uh, we won't have any drug left for E. coli also. Uh, Staphylococcus aureus, another infection which is the commensal bacteria in the skin, and it also caused many infections in community. And the penicillin and then methicillin was sensitive, but now 64% of the population are more likely with the methicillin resistant staphylococcus infection, and this leads to the spread of the infection with the Staphylococcus aureus. Another important bacteria is the Mycobacterium tuberculosis, which is the causative factor. And if we see the 2018 data, then you can see that tuberculosis is a very common global epidemic, and WHO estimates that about 500,000 new cases of rifampicin resistant TB and multi-drug resistant TB identified. Tuberculosis, rifampicin is the first line drug, and if this drug become resistant, and also the multi-drug resistant, which is having in multi-drug resistant TB, there are at least two drugs which are resistant. So there are cases where we have only rifampicin, and in some conditions they are multi-drug resistant TB. About 500,000 new cases are found, and only one third of approximately 500,000 people who develop multi-drug resistant and rifampicin resistant TB, they are detected and reported. So one third of the 500,000 people are detected, and estimately 3.4% of new TB cases and 18% of previously treated cases had multiple drug resistant and rifampicin resistant tuberculosis. So you can imagine that if a person is suffering from multi-drug resistant tuberculosis, it requires treatment for long period of time, and less effective and far more expensive than those for non-resistant tuberculosis. And less than 60% of the patient treated with MDR and rifampicin resistant TB successfully cured. So the emergence of even the new last resort TB drugs may lead to the major threat to in the treatment of tuberculosis. So after seeing the resistance in bacteria, resistance also found in viruses, and especially in conditions where the person is immunocompromised and where the viral replication is more in some viruses, and prolonged drug exposure may lead to the resistant strains in the viruses. Uh, this is more commonly seen in the use of antiretroviral drugs in the HIV/AIDS patients. So in the HIV/AIDS patients, patients are becoming resistant to even the newer class of antiretroviral, and they are called as HIV-DR, or the drug resistant TB. So people who are receiving the drugs, they can acquire this HIV-DR or drug resistant HIV. So people are becoming more and more sufferer of this drug-resistant HIV. And these patients, if they are pre-treated with non-nucleoside reverse transcriptase inhibitors as an initial first-line therapy, so this also exceeded to 10% or in majority of the monitored where this program is being monitored, especially in Africa, Asia, and America. So the prevalence of the pre-treated HIV/AIDS is more in infants, and it's alarming. And over 50% of infants newly HIV diagnosed carry the virus resistant to non-nucleoside reverse transcript inhibitors. So in the population where people are becoming resistant even to NNRTI, then WHO suggested to use dolutegravir as a preferred first-line treatment for adults and children because this may cause less resistance to the NNRTI. And because of this resistance, WHO is working on HIV drug resistance program and monitoring the whole program globally. Then we can also see that drug resistance is also found in a common disease, malaria. I'm just discussing tuberculosis, malaria, and HIV because these are the very common and diseases. And if we have the resistance in the drugs which are used in tuberculosis, HIV, then it will cause more problem. So this is something about the resistant malarial parasite, and most of the WHO regional offices, they are reporting that there is now artemisinin combination therapy resistance in most of the countries. Like you can see like Cambodia, Laos, Myanmar, Thailand, they found that there is a partial resistance to artemisinin as well as artemisinin combination therapy. And in some the Eastern Mediterranean region, Plasmodium falciparum found resistant to sulfadoxine pyrimethine. Then it led to the failure of the artemisinin combination therapy with toxin and pyrimethine failure, and this leads to the change in another artemisinin combination therapy. So artemisinin can be combined with other the partner drugs, and point is how far we can change or modify the artemisinin combination therapy when we will not have any ACTs left. So, drug resistance is found in bacteria, viruses, and some protozoas, and also in fungi. But in fungus, we all know that usually fungal infection is difficult to treat, especially the fungal medicines are very toxic and toxic, as well as when a person is having comorbid conditions like HIV, where the person is having the fungal infection as well as HIV. So if we are having the resistant antifungal drug, then it will be difficult to treat the fungal infections with the comorbid conditions. Similarly, Candida auris, which is a common invasive fungal infection, this is also found resistant to most of the antifungal drugs like fluconazole, amphotericin B, voriconazole, and also caspofungin. So this led to the more difficult in the treatment of fungal infections, treatment failures, longer hospital stays, and much more expensive treatment if we are having resistant common antifungal drugs. So WHO is undertaking a very comprehensive review of the fungal infections at a global level, and sooner or later WHO is going to publish a list of fungal pathogens of public health importance along with an analysis of the antifungal development which is in the pipeline. So after seeing this problem in the bacteria, in the viruses, in the fungal, and some protozoal, then we need to have prevention and control strategies, which is done through the through global efforts. And a problem is where we don't have proper infection prevention and control measures. So this prevention and control measures should be taken at all levels of the society to reduce the impact of the antimicrobial resistance and limit the spread of the antimicrobial resistance. And these steps need to be taken by individuals, by policymakers, by health professionals, by healthcare industry, as well as veterinary and agricultural sectors, because these are the stakeholders of the antibiotics. So let's see how individual can help in control and and prevention of the antibiotic resistance. And individuals are the most important stakeholder, and they should have more responsibility in prevention and control of the antibiotics. So they are supposed to use antibiotics when they are asked by certified health professionals. And it is found that consumers and the patients, they demand antibiotics to be prescribed by health workers, and they insist that antibiotics should be there in their prescription. So never demand antibiotics if health worker does not suggest for you any antibiotics. Always follow the health worker's advice. Never share or use leftover antibiotics. These are the common mentality in India, that we usually share and don't leave the antibiotics. So leftover medications are another problem in the use of antibiotics. Then we need to prevent infection by other measures. Antibiotics are the last resort. It's better if we regularly wash, prepare food hygienically, avoid close contact with sick people, practicing safer sex, and keeping vaccinations up to date. So this may prevent infections if we follow these simple guidelines. And WHO specifically given some guidelines for preparing foods. These are the 5 keys to safe food. So it's keep your food clean, separate raw and cooked. Third is cook thoroughly. Fourth is keep food at safe temperatures. Fifth is use safe water and raw materials. And again, you need to choose the foods, if you are going to buy from the market, that have been produced without the use of antibiotics. In some countries, it is already mentioned in some food that this is without the use of antibiotics. And you know that antibiotics are used in the food for growth promotion or disease prevention, even in healthy animals. So another stakeholder are the policy makers, and these policy makers, they can also play a role to prevent and control the spread of infection. They are supposed to ensure a robust national action plan as per the WHO Global Action Plan to tackle the antibiotic and antimicrobial resistance, and to improve the surveillance of antibiotic resistant infection. You know that in almost all the hospitals, and even at the national level, we have surveillance team for monitoring the antibiotic resistant infection. So policymakers need to improve the surveillance team where it is not existent. Strengthen the policies, programs, and implementation of infection prevention and control measures. Programs like the antimicrobial awareness program, or the WHO programs, which is there every year. Regulate and promote appropriate use and disposal of quality medicines. Make information available on the impact of antibiotic resistance. Then third stakeholders are the health professionals, because they are going to prescribe and most of the time dispensing the drugs. So they can prevent infection by ensuring to clean and wash hands, and wash instruments and environment. They keep the environment clean where they are practicing, or environment clean clinical setup or hospital. Prescribe and dispense only the antibiotics when they are needed, and or as per the current standard treatment guidelines. because these standard treatment guidelines are made in consensus with the hospital where we have clinicians and then microbiologists and pharmacologists. And if there is any antibiotic resistant infection, that needs to be reported to the surveillance team. And as I said, the surveillance teams are there in most of the hospitals. Every patient should be explained how to take antibiotics correctly and about the antibiotic resistance, the possibility of antibiotic resistance, and the dangers of misuse. These health professionals need to guide patients about preventing infections through vaccination, hand washing, safe for sex, and covering nose and mouth when sneezing. So these should be told to the patient also. Healthcare industries can also play a role in the control and prevention of the spread of antibiotics in investing in research and development of new antibiotics, vaccines, diagnostics, and other tools. So these are the stakeholders. And last stakeholder is are the veterinary and agricultural sector, where antibiotics are commonly are becoming used. They are told that they need to give antibiotics to animals under the veterinary supervisions. So only when veterinary doctors allow to give antibiotics, then they need to give in the animals. And never use antibiotics for growth promotion or to prevent diseases in healthy animals, because this is the sector where we are witnessing the more number of antibiotics used in the poultry farm, where antibiotics are given for the growth promotion, hormones are given for the growth promotion. So it needs to have vaccination to animals rather than the use of antibiotics, and also need to have alternatives to antibiotics when available. Promote and apply good practices at all steps of the production of the poultry farm or aquatic farm or fisheries, wherever you are using the medicines, and also in while processing of foods from animals and plant sources. So there is a need of improving the biosecurity on farms and prevent infections through improved hygiene and animal welfare. Now, after seeing the resistance in various microbes and the how we can prevent, and even if we can't prevent the infection and control the infection, then there will be a huge impact of this antibiotic resistance in the society. And this may lead to the use of second line antibiotics when first line antibiotics become resistant. And these second or third line antibiotics, they are more expensive. They compromise the ability to treat the common infectious diseases. And these resistant bacteria, they need to be treated for long period of time, and they also need hospital stay. So there is an economic burden also in the healthcare cost and on families and societies. And ultimately less treatment, and then may lead to increase in morbidity and mortality. So antibiotic resistance, if there, because in allopathy, in modern medicine, antibiotic is the most important and armamentarium in the pharmacology. So if the antibiotic resistance is there or increases, then it will increase the risk of the achievements in the modern medicine, and undermine the advances in health and medicine, especially in the areas where organ transplantation, chemotherapy, and surgeries where cesarean sections become much more dangerous without effective antibiotics for prevention and treatment of infections. So if we continue to witness the antibiotic resistance, then we will go to an era which will be called as post-antibiotic era. Like we had pre-antibiotic era, where there was more casualties because of the non-availability of antibiotics. And this post-antibiotic era may lead to the same situation, where we will not have the treatment even for the minor injuries, and this may lead to the death. So WHO has come up and is playing an important role in the global response of the antibiotic resistance. resistance, and it's a priority program to tackle the antibiotic resistance for WHO. And in 2015, WHO, with the endorsement of UNO and other bodies, started to have antimicrobial resistance program for the containment of antimicrobial resistance in the Global Action Program came in 2015, but the Global Strategy for Containment of Antimicrobial Resistance came in 2001. So it provides a framework of intervention to slow emergence and reduce the spread of antimicrobial resistance. As I said, in 2015, Global Action Plan came up during the 68th World Health Assembly, where they endorsed this Global Action Program, including the antibiotic resistance. And the WHO supporting member states, they adopted this Global Action Program. And the goal of the draft Global Action Program was to ensure for as long as possible the continuity of successful treatment and prevention of infectious diseases with effective and safe medicine that are quality assured. Because as you know that since 1960s, there was a problem of antimicrobial resistance, but this is the concerted program started in 2011 and more after 2015. So this global action program on antimicrobial resistance has five objectives, and these five objectives are to improve awareness and understanding of antimicrobial resistance. So WHO, for this strategy, for this objective, there are like World Micro Antimicrobial Awareness Program, which is celebrated in the month of November to strengthen knowledge through surveillance and research. Again, another program for this activity. Surveillance teams are there, research activities are there to reduce the incidence of infections. That's why hospital infection society or hospital infection measures are there in each hospital to minimize the use of antimicrobial agents through standard treatment guidelines and to develop economic case for sustainable investment that takes account of the needs of all countries and increase the investment in new medicines through raising funds. And so these can be achieved in developing new medicine, diagnostic tools, vaccines, and other interventions. So to fulfill these strategic objectives, WHO has other programs. So India is also a signatory of the Global Action Program on Antimicrobial Resistance, and finally, the National Action Plan on Antimicrobial Resistance came in 2017, in the April 2017, and it's a part to ensure global progress because each country needs to ensure costing and implementation of national action plans across sectors to ensure sustainable progress. So this antimicrobial resistance program is also having One Health Global Leaders Group. So to fulfill these global action programs, we have some strategies. One strategy is One Health Global Leaders Group, and you know that WHO in association with the UN, uh, FAO, Food and Agriculture Organization, World Organization for Animal Health, they signed and declared and endorsed this One Health Global Leaders Group in September 2016. And this led to the signal to the world commitment to taking the broad coordinated approach to address the root causes of antimicrobial resistance across multiple sectors. We have seen that antibiotics are misused in multiple sectors, especially in human population, in animal population, and in agricultural setup. So these are the coordinated approaches to address the root cause of antimicrobial resistance in these areas. And this is also called as tripartite program for One Health Global because the main agencies are the FAO, Food and Agriculture Organization, World Organization for Animal Health, and WHO. So they are promoting best practices to reduce the levels of antimicrobial resistance and slow its development. So this new global leader group will advance the global action program and ask different countries to adopt the national antimicrobial programs on in each country to ensure and to generate in generations to come, we can continue to prevent and treat infectious diseases with safe and effective medicines. So this is One Health program, and for this One Health program, we have the Interagency Coordination Group, which is called as IACG. And again, as I said, it is the across the UN and international organizations and individuals, and the purpose is to fight against antimicrobial resistance and to ensure effective global action against this threat. So this IACG is convened by the Secretary General of the UN, and they are also going to implement in the world about this report. No time to wait, securing the future from drug-resistant infections. So this is in diagrammatically, you can see that in the One Health initiative, the if we protect the humans and environment and animals, then this leads to the animal environment human interface and One Health concept. Likewise, here you can see that it's a coordination, communication, and collaborating between these animals, humans, and environment. So it's basically ecosystem which needs to be free from the bacteria. Uh, so critical importance of One Health approach to antimicrobial resistance is like we know that antibiotics are used in human as a therapeutic indication and in hospitals as well as in community setup, and this leads to the resistant bacteria. These bacteria may reach to the environment, and similarly resistant bacteria may come into the environment from animals, from plant, from agriculture, and there is an interlinking of this, and resistant bacteria may reach into the environment and may reach to the lakes, rivers, and soils. And you can imagine that when vegetables are grown in such type of soils, then and the water and from agriculture and from vegetables, the resistant bacteria may reach to the human, and antibiotics also are used in pharmaceutical industries. They are also thrown and sometimes antibiotics are thrown in the soil and rivers. So you can imagine in our diet, in our water, and resistant bacteria may come from different sources. So there is a need to check all the steps so that lesser resistant bacteria may reach to the human health. So there's something again the same thing like there is a need for coordinated action as WHO has taken with the help of FAO and World Organization for Animal Welfare. And second thing is there is a launch of antimicrobial resistance multi-partner trust fund, because funds are also needed to curb this menace and to successfully organize this global action program. So there is Global Antibiotic Research and Development Partnership, there is Antimicrobial Resistance Action Fund, and other funds and initiatives that could bridge a funding gap in running these programs. And many national governments, agencies, they have already adopted some models to run this coordinated action program. So another coordinated effort is the formation of a global antimicrobial resistance surveillance system. As I said, surveillance system is there in each hospital, but global level, this is called as GLASS. And many antibiotic resistance pattern are being shared. The data are being shared at global level, and then GLAS may share the data to the nation, to different countries. So this is GLAS, which was launched in 2015 to continue filling the knowledge gaps and to inform strategies at all levels. So this is based on the surveillance data which comes from different countries, and it helps in knowing the status of existing antimicrobial resistance and in framing the new national surveillance system, and with emphasis on representativeness and quality of data collection. So this is maintained by WHO, the GLAS. Another strategy is having GARDP, which is the Global Antibiotic Research and Development Partnership. So, with this private industry partnership, uh, this leads to the research and development into the synthesis of new antimicrobials, new diagnostics, and new vaccines. And this is also WHO priority, and WHO has also framed priority pathogens list. And on annual basis, they review these research and development, and they try to update every 2 years. So another updated list will come in 2020. And with this GARDP, WHO has signed with the another program, Drugs for Neglected Diseases Initiative, because Drugs for Neglected Disease Initiative, which is DNDI, also needs to have newer drugs and research. So this GRDP encourages research and development through public-private partnership, and purpose is to have new treatment strategies and improvement of the existing antibiotics and accelerating the entry of new antibiotic drugs. So GARDP is another good step by WHO, and this is WHO priority list where they are asking GRDP to work on, and you can see that in the priority list, some antibiotics resistant with some resistant bacteria and resistant antibiotics. These are priority 1, priority 2, priority 3. So these are critical. These are priority 2 or high. This week priority list. So you can imagine, see that WHO is working on and gives list of priority list for research and development. Another strategy is the World Antimicrobial Awareness Week, and every year from 18 to 24 November, there is a World Antimicrobial Awareness Week, and the purpose is to create awareness in the general public as well as in the doctors about the antimicrobial resistance problem, and to encourage best practices among the general public, health workers, and policymakers to avoid further emergence and spread of drug resistant infection. So this has been observing since 2015. So it's a very critical tool in helping to fight diseases in humans, animals, and plants. And we can also discuss One Health concept during the World Antimicrobial Awareness Week. And so, like World Health Day theme, there is World Antimicrobial Awareness theme also. Like for example, earlier the antibiotic handle with care was the theme, and now in last year it has been antimicrobials handled with care. From antibiotics has been changed to antimicrobial to reflect the broadening scope of drug resistance, because there is not a problem of antibiotics but a problem in the virals and in the bacteria. So this is a part of World Week. Every institutions, organization, they observe World Antimicrobial Awareness Week. This is something about the they observe the World Antimicrobial Awareness Week, and these are some posters and which are circulated by WHO during the World Antimicrobial Awareness Week, and in different languages and in different situations. Now come to the second part of my presentation, which I would like to cover within few slides or in short. So mechanism of the drug resistance. So you know that antibiotic resistance. Would you like to break or we should continue?
Please continue, sir.
Bilal?
Please continue.
Oh, okay, fine. Okay,
Continue,
Bilal.
continue, sir. Continue, sir.
Well.
Continue, sir. Thank you.
So antibiotic resistance, is developed by bacteria in the couple of hours, and it provides the ample opportunity for genetic adaptation. And this resistance is found through the natural way or genetic way or acquired way. So genetically is through mutations and through extra chromosomal mechanism plasmids. So if we see the chromosomal methods, then bacteria, they develop spontaneous mutation, and this occurs at a frequency of approximately 1 per million cells. And although mutation rate is slow and there is a selection of mutants, and which may lead to the reduced pathogenicity, but is more problem. The spontaneous mutation is more problem if we are having common diseases like tuberculosis and leprosy, especially methicillin resistant staph aureus. And these are some examples like streptomycin and rifampicin. They became resistant with this single step mutation through multiple step mutation, erythromycin, tetracycline, and chloramphenicol. And this is that was chromosomal method. This is non-chromosomal method, which is using plasmids. And so this plasmid, this is also called as infectious resistance. So, you know, plasmids, they carry genes, resistant genes, which is called as R genes. And these R genes are transferred from one microorganism to another organism. That is why it is called as infectious resistance. And this R genes are transferred to another bacteria through gene conjugation, through gene transduction, and through gene transformation. So there are three ways of the gene transfer: conjugation, transduction, and transformation. So conjugation is where it needs physical contact, and physical contact through pili, which is the sex pili, and this physical contact then able to transfer the plasmids having containing the R genes from one bacteria to another bacteria. And it's a main mechanism of the spread of the resistance, especially in the where the bacteria density is high, as in gut. And this is achieved in the development of resistance against streptomycin and chloramphenicol. So this is gene conjugation, and another is gene transduction. And this gene transduction is through plasmid enclosed in the bacterial viruses, which is called as bacteriophage. So bacteriophage is responsible for the carrying the R gene from one bacteria to another bacteria, and this leads to the another bacteria for the with the resistant genes. So this is found in strains of Staphylococcus and in Streptococcus. And when these strains become resistant, then these drugs like penicillin, erythromycin, and chloramphenicol are difficult to give. So this is a way of inter exchange of resistance. For example, Staphylococci may transfer genes to Enterococci, and likewise, so spread may be there to other bacteria also, not only from one bacteria to another, but from one bacteria to another bacteria. So this is gene transformation. Third is gene transformation. Although this is least clinical, it causes least clinical problem, but here genes are transferred freely, and this is the recipient from one environment to the closely related of the or the same strain. So new DNA incorporated into the genome of the bacteria, which become the resistant. So this is found in penicillin G. So these transfer of R genes occurs through transposons. Transposons are the DNA segments that cannot self-replicate but can self-transfer between plasmids or from plasmids to chromosomes. Donor plasmid contain transposon and it co-integrates with the acceptor plasmid, and this way it helps in the transfer. Integrons also play roles like transposons in transferring the resistant genes, and this integrons they spread by larger mobile DNA, each integron packed with multiple gene cassettes. And these gene cassettes, multiple gene cassettes consist of resistant genes attached to a small recognition site, and these multiple gene cassettes encode several bacterial functions including resistance and virulence. So currently gene cassettes identified for all antibiotics except fluoroquinolones. Uh, Second mechanism is the biochemical mechanism, which is basically the elaboration of enzymes by bacteria. So these enzymes elaboration, they inactivate the drugs. Like for example, acetyl transferase is overexpressed in some bacteria. This leads to the inactivation of chloramphenicol. Beta lactamases is overexpressed in some bacteria, especially inactivate the beta-lactam antibiotics like penicillin, cephalosporin, carbapenem, acetyl transferase, phosphotransferase, and adenyl transferase. They may lead to the inactivation of aminoglycosides. So elaboration of enzyme in another mechanism. Third mechanism is the decreased influx. So when there is decreased influx by bacteria, then antibiotic may not reach into the bacterial cell, and which is also responsible for the inactivation of drug. So this may be because of the uh, inhibition of porin channels and decreased drug permeability due to loss of specific channels like aminoglycosides and tetracyclines attain much lower concentration in resistant organism than in the sensitive organism. So these needs to be entered into the cell through porins or the so there is less influx. And on the other side, there may be promotion of efflux pumps. So when efflux pumps are more active or overexpressed, then they also lead to the expulsion of antibiotics from the cell, and they don't allow the entry of the antibiotics and work as a or does not allow its antibiotic activity. So development of efflux pumps are seen in tetracyclines, erythromycin, fluoroquinolones, and active so this leads to the extrusion of drugs. And another mechanism is modification and protection of target site. We know that antibiotics, they act on certain targets. For example, the ribosomes. So ribosomal point mutations are seen with drugs like tetracycline, macrolides, and clindamycin because they act on they inhibit the protein synthesis on action on ribosomes. So there is a change of the amino acids in the subunits of DNA directed RNA polymerase in the mycobacterium. So we know that rifampicin acts on the inhibition of RNA polymerase, and if there is mutation of this enzyme because of the ribosomal role, altered DNA gyrase or altered topoisomerase. This also leads to the non-functional fluoroquinolones, altered penicillin binding proteins in pneumococci and staphylococci. So these are the target site ribosomes. Could be the enzymes, could be enzymes like DNA gyrase, topoisomerase, and the penicillin binding proteins, which is in short called as PBP. Sometimes bacteria they prepare alternative metabolic pathway for its survival. We know that folic acid is synthesized from PABA, and when we inhibit the step or the conversion of folic acid from PABA, through giving drugs like sulfonamides and cotrimoxazole or trimethoprim. So what bacteria started doing, they developed the alternative metabolic pathway. Instead of getting folic acid from PABA, they may use preformed folic acid, or they may overproduce the PABA so that they can get folic acid for its survival. So, or overproduction of beta lactamases, which inactivates the drug. Another mechanism is quorum sensing, which you most probably know that quorum sensing is very important and recently discovered that regulation of gene expression is in response to fluctuations in cell population density. So whenever there is increase in the cell density population, so they started regulating gene expression. So quorum sensing bacteria, they produce and release chemical molecules whenever they have cell population density, and whenever there is increase in concentration as a function of cell density. Detection of minimal threshold stimulatory concentration of an autoinducer also led to alteration in gene expression. So both gram-positive and gram-negative bacteria, they use quorum sensing to communicate and regulate the physiological activities. And these physiological activities through various processes like symbiosis, like virulence, competence, conjugation. So all these physiological processes are governed through the quorum sensing, sporulation, motility, and most importantly, biofilm formation, because nowadays most of the antibiotics are being targeted on biofilm formation. So in general, gram-negative bacteria use acylated homoserine lactones as autoinducers, and in gram-positive bacteria, this is oligopeptides to communicate and to overexpress the autoinducers. So this is quorum sensing. So the purpose of quorum sensing is making the biofilm or protective layer, and this is because of development of multicellularity, and number of compounds, as I said, have been synthesized, which inhibit the quantum quorum sensing system, which includes the enzymes like AHL, as we have seen that it's common in is responsible for in the gram-positive bacteria for making the quorum sensing. Then resistance, if we have seen that there are many mechanism of resistance, like genetic, like enzyme, like quorum sensing. Let's see a few examples of resistance in some specific microbes. So resistance in macrolides and lincosamides and streptogramins, they are inherited together, which is called as MLS. M is macrolides, L for lincosamides, and as is streptogramins. So this is resistance to these. This is responsible for the three antibiotic resistance. Clindamycin resistance is because of the alteration of the 50S ribosomal subunit by adenine methylation, and also by chromosomal mutation of 50S ribosome, and also plasmid mediated aryl cycle transferase. So each antibiotic may have different mechanism of antibiotic resistance. How sulfonamide are become resistant to to bacteria like gonococci, staphylococci, meningococci, streptococci, and this is because of the mutations in the bacteria, and may lead to overproduction of the PABA to overpower the inhibition due to sulfonamide. We know that sulfonamide inhibits the steps of synthesis of folic acid from PABA. So alteration in the nature of dihydroxypyroic acid synthetase. So this alteration may also lead to the low affinity of for the sulfonamide, the loss of permeability through bacterial membrane, appearance of alternative pathway for PABA. Alternative pathway is preformed folic acid from some other source, not from the PABA. Resistance to penicillin is very commonly seen and very much discussed, that the penicillin is becoming resistant even to gram-positive bacteria, and this is because of the development of penicillinase in the bacteria or altered penicillin binding proteins. And to minimize the penicillin resistance, we were able to develop beta lactamase inhibitors, which inhibits the beta lactamase enzyme, which is produced by bacteria. And they came up with penicillinase resistant penicillins like cloxacillin, oxacillin, nafcillin, dicloxacillin, and methicillin. And now we are also developing, we are seeing the resistance with these beta lactamase inhibitors. For example, and this is called as extended spectrum beta lactamases. So this extended beta lactamases covers not only penicillins but also cephalosporins and monobacteriums. And extended spectrum beta lactamase found exclusively in gram-negative organisms, primarily in Klebsiella and E. coli. Important characteristics of this extended spectrum beta lactamase is because of the functional genes like Bush 2BE and molecular group Amber group A. And likewise, we are also metalloproteinases variants which have NDM1, NDM2, NDM2, 3, 4 variant, and this is also creating problem if we have NDM, because this may lead to the resistance even in the most important and potent antibiotics like carbapenem. So aminoglycosides also having resistance because of the inactivating enzymes like aminoglycoside, acyl transferase, phosphotransferase, adenyl transferase, and these enzymes, when overexpressed, they may metabolize aminoglycosides and inactivates the aminoglycosides. So all aminoglycosides except amikacin and nettlemycin are susceptible to these or enzyme, and so nettlemycin and amikacin may be effective against organism resistant to other aminoglycosides. And apart from this plasmid mediated enzymatic modification, other mechanism of resistance in aminoglycosides is because of efflux mediated resistance, which is the mutation in purine channel and deletion or alteration of receptor protein on 30S ribosomal unit. Tetracycline resistance is because of the development of efflux pump, because of the decreased cell permeability, because of ribosomal protection, because of enzymatic inactivation. And chloramphenicol, again, there are many mechanisms: reduced morbid membrane permeability, mutation of the 550S ribosomal subunit, elaboration of the enzyme CPL, this chloramphenicol acetyl transferase. And this chloramphenicol resistance also found through plasmid that code for resistance to other drugs, which is called as ACCOT. A for ampicillin, C for chloramphenicol, and CO is cotrimoxazole, and T is tetracycline. So this shear the resistance in all these antibiotics. Um, Enterococcus faecium and Pseudomonas aeruginosa strains, they are becoming resistant to chloramphenicol, and Vinola species and Staphylococcus also becoming resistant to chloramphenicol to varying degrees. And because of this, we have rapid uh, high toxicity and fewer systemic effects. Now, in the last, we will, because tuberculosis is also very important area facing difficulty in the treatment, and this because of the resistance of the drugs like resistance in rifampicin. And in rifampicin, it is found that bacteria, they are responsible to inactivate the drugs because of the RNA polymerase, um, uh, which is the gene is RPOB, and alternative mechanism through ARR catalyze ADP ribosomal ribosylation of the rifampicin, Mycobacterium sphagmatis. Again, this is an enzyme ARA is found. So rifampicin, pyrazinamide, again we can see there are mutations in the treatment with pyrazinamide, and these are some examples of the gene mutations in the RPSA and streptomycin is another group of drug. So rifampicin, isoniazid, streptomycin, and streptomycin point mutation because the RP cell or RRS gene. So this is all in short about the antibiotic resistance and how antibiotic resistance develop and in the last, I would like to list that there are many publications by many experts who are working on antimicrobial resistance. Many labs they are working on antimicrobial resistance, and there is a long list of WHO publications, but these are two very common publications which you can refer. So thank you so much. We have come to started from the Alexander Fleming penicillin and which he first developed penicillin, and before, so starting from the pre-antibiotic era, we are uh, furthering for in the era of post-antibiotic. So it's a concern area. So once again, thank you so much, and I would love to have some questions if there is some.
Thank you very much, sir. Thank you very much, Professor Said Ziaur Rahman, sir. So now we have, I think, if the presentation is over, now we have some questions. That the floor is open for
Yeah.
the questions. Let me first read some questions that have been already posted on the chat box. So there are some questions that have been posed by the participants on the chat box. The first question that has been posted by Dr. S.M. Zubair. Does antibiotic
Mhm.
resistance, especially multi-drug resistance, increase vulnerability of a patient to varied diseases? If so, what is the way out as only specific antibiotics are recommended to such patients with MDR?
Hmm. So, as we discussed, there is cross resistance is found. So, cross resistance from one bacteria to another, and if and this cross resistance as well as the resistance
You are not audible, sir. Your voice is cut.
we need to have strategies to prevent and control the diseases. So again, we have already discussed lots of strategies in the prevention of such type of condition. I think this is a question which is something which is needs. It's a gist of the whole lecture which we covered. So the whole lecture gives the answer of this question.
Thank you very much, sir. So the another question the same person has asked posed here is how can we contain AMR, antimicrobial resistance?
See, to control antimicrobial resistance, there are many strategies. As you can see that WHO has framed the antimicrobial resistance policy and global action plan. And these global action plan, they are asking different countries to have national action plan. And in national action plan, we have already seen that there are various steps. Uh, we are raising funds. We are asking industries to work on newer antibiotics. We are asking different stakeholders to minimize the use of antibiotics. Uh, uh, we are asking uh, so many stakeholders to minimize its use. Uh, so this may lead to the inhibition of the use of antimicrobial resistance through various stakeholders.
So we have another question from the participant, Dr. Mushtaq Ahmad Tantre. He is posing that presuming antimicrobial resistance continues at a present rate, and God forbid if no newer classes of antibiotics are found in next decade, what could this situation lead to, particularly in developing or underdeveloped countries? Is the fear mentioned in various WHO reports completely facing us?
Yes, as I said, this may lead to the post-antibiotic era. So, pre-antibiotic era, the problem was when we had First World War and Second World War, there were many soldiers, they were lying uncared, they were lying with wound injuries, and these wound injuries leads to the bloodstream infection and death. So this led to the development of antibiotics, and luckily we had penicillins and then sulfonamides and other antibiotics which came up and helped in the treatment of various infections. If we are having this resistance problem, and if this is not solved immediately, then we will go back to the same situation, which will be called as post-antibiotic era. And in post-antibiotic era, we will not have any antibiotic left, because we just now we have seen that some infections, they are cured by first line antibiotic. If they are becoming resistant, then we go for the second line antibiotic, then third line. And you know that these newer drugs, they are more expensive, they are not cost effective, and they are less effective also. So this may lead to the more vulnerability to a more serious infection.
Okay, sir. The next question, which is posed by Dr. Manzoor Ahmad Mir. Sir, is combination therapy the only possible solution to drug resistance? Should we go for developing new drugs or use the already available ones in combination? Please give your
Yes.
inputs.
No, in some conditions, we don't require combination therapy. In some conditions, we require combination therapy. So it is the responsibility of the doctors, and also it should be mentioned in the standard treatment guidelines in which conditions there is a need of combination therapy and where combination therapy are not required. So combination therapy is basically purpose is to minimize the resistance, to increase the efficacy, and mainly there are two reasons to increase the efficacy and to minimize the resistance. So combination therapy are required in some condition but not in most of the condition. uh, uh, Combination therapy uh, uh, serve a purpose, but not always. So we can't say that in all the conditions combination therapy are used and it will help in the decrease of the resistance. So it depends on the conditions and the disease. As I said, it's all should be mentioned in the standard treatment guidelines. So, like we have standard treatment guidelines in most of the Delhi hospitals. So it's basically prepare after consultation with the hospital infection society, or after seeing the pattern of the antibiotic sensitivity, then they can decide which combination should be used, which should not be used. For example, we have seen that in leprosy, in tuberculosis, these are the conditions where we need to have combination therapy.
Sir, thank you very much. There is another question from Dr. Siraj Pare. He says that can we block ABC proteins in cells, thereby making cells susceptible to antibiotics wherever needed?
Yes, there are strategies. Um, we can block this ABC also. We can block the enzymes which are overproduced by the bacteria. We can block the quorum sensing strategies, like we can block biofilms. We can block some enzymes which helps in the release of toxins. uh, there are now we know the mechanism of resistance, so we can work on different areas where we can inhibit the mechanism of resistance. If we know the mechanism of resistance, then we can proceed in the development of new drug and check the resistance. But this requires lots of funds, lots of research. and many laboratories they are working on these strategies which you have mentioned.
So the next question from Shaukat Ahmad is: as far as AMR is concerned, what is the scope of products obtained from medicinal plants?
Um, Yes, they are claimed by traditional practitioners, and there are some researches also that plants may have antibacterial activity, but they are still under pipeline, and but very few drugs like anti-cancer drugs they have shown chemotherapeutic effect, but um, probably yes, because nature has provided cure for every disease. So yes, probably yes, we can have in near future antibiotics from the herbal sources, but still we are not sure or can't comment because as we had discussed also in our presentation, modern medicine or allopathy is proud to have this antibiotic, and most of the antibiotics they are derived from the fungus or bacteria or synthetic. So nowadays, most of the antibiotics they are synthesized using the biotechnology and synthetically. But from plants, yes, we can have yes, in future. Yes, we
Okay.
can't say, but it's still. But at present, there is no drug which is derived from the mainly from plants.
So the next question from Shaukat Ganai. I think it should be Professor Shaukat Ganai from biochemistry. Please explain how the bacteria reduces the effectiveness of the drug.
Reduce the effectiveness of drugs. So we have
Effectiveness of the
already,
drug.
yes, we already discussed lots of mechanism
Okay.
in reducing the effectiveness of the drug. We have seen that um, uh, this is the smartness of the bacteria that they develop some mechanism to inhibit the effectiveness of drug because that is why they are called as superbugs. They are very smart, it's smarter than us, and they learn how to survive and adapt the environment. Many mechanisms are there with which the bacteria survive to live.
The next question is: how can we contain decreased influx of drugs?
Decrease the influx of drug.
Influx of drugs.
Uh, uh,
Contain the decreased influx of drugs. It, it is
Influx
it
to
is
where? Influx to
it is it is basically to contain the decreased influx of drugs. It is the question is about how
okay.
can we contain the decreased influx of drugs.
If it is concerned with the efflux or influx of drug in the bacterial cell, then we know that drugs are permeable into the bacterial cell through certain porins or channels. And we have also seen that some bacteria they increase the efflux system and decreases the influx system. So many scientists are nowadays working on these porins and uh, try to close or block these porins. So there are drugs which are under pipeline which is helping in the blockage of these porins, and this will help in the decrease in the efflux and increase in the influx of drugs into the cell.
So the next question is: co-evolution cannot be stopped. What is the way out?
Cool.
It is co-evolution cannot be stopped. What is the way out? It is posed by Shaukat Ahmad.
Uh, See, this is ecosystem where we are living. We cannot protect only humans. That is why One Health concept came and One Health initiative was taken by WHO. There should be concerted efforts to protect the environment, to protect the animals, and to protect the human. So if we live together, or if there is a healthy coexistence, then we can live together. Or so coexistence in terms of healthy environment, healthy humans, healthy animal is very important. Like we have seen that we are polluting the environment through antibiotics and the antibiotics, they are reaching into the aquatic environment, they are reaching into the terrestrial environment, and there is a misuse of antibiotics in the aquaculture and in poultry and in fisheries and in growing vegetables, and also sometimes they are giving drugs or antibiotics in the healthy animals. So point is for coexistence, for the survival of each individual, there is a need. That is why WHO came up with the concept of One Health. So we are not talking about the health of the human, but also talking about the health of animals, talking about the health of environment. So these three are, if these three are healthy, then this will lead to the One Health concept, and for the all these.
So, that Jyoti Taneja has a question, or rather, she has a query. What about unused antibiotics, remaining dose, and
Hmm.
thrown directly in waste bins?
Yes, there are many programs for unused antibiotics. In some cities, there are depot where unused medicine should be deposited, and there are some guidelines also for the unused or leftover medicines. Sometimes unused or leftover medicines, they are incinerated and they are converted into the biodegradable form. So they are sometimes taken as a bio waste material, and very cautiously they are removed from the environment. So there are programs for leftover unused medicines, and it's better if we don't have, if we don't know anything about these programs, then it's better to discuss this problem with from the pharmacist from where you got the drug, or pharmacist may help in disposal of these drugs, or if these leftover medicines, they still have the shelf life, they can be reused, but not should not be used means one or two drugs only. So proper dose should be utilized.
Another question, I think even microbes are found having colistin resistance lately. What
Hmm.
are the options left to combat the antibiotic resistance
Very
problem?
difficult, difficult situation when we have colistin resistant or carbapenem resistant, because these are the last resort for most of the patients. If we have then uh, only mortality is left. We have been seeing that most of the infections are untreatable because of these colistin and carbapenem resistant areas.
The next question is, can we map cell surface receptors on all human pathogens and develop broad spectrum vaccine against human pathogens?
Possibility is there, yes, we can't say, but yes, it's a good question, and it's a good question for research and development. We can have, yes, we can synthesize drugs, and this idea should be floated to um, uh, researchers.
I think all questions. There are so many other
Bilal
questions,
sir,
and
Bilal sir, I have a question. Can I ask?
Yeah, sure, sure.
let me finish. Let me finish the questions that
Okay.
have been already posted on the chat box. There's one more question from Dr. Tariq Makbul. He's saying, what is the way forward? Can alternative sources or new approaches like nanomedicine and nanotechnology help? Any update, please?
Hmm. No, there are papers. Antibiotics are being tried through nanotechnology, and there are papers where and newer way of administering drugs are there. But still, they are not commonly used or came up in the market. But I heard and I saw some papers which are working, but they are not very successful. Still, they are not successful.
I think we have many few questions, but due to the constraint and time, so if anybody, we have a few
Yes.
minutes
Can
left.
I
If anybody
can I
wants to
ask you a question,
yes,
Bilal
you
Sahab?
can. Yes, yes, you can go ahead.
First of all, thank you, Professor Ziaur Rahman Sahab. A very important lecture. Want to have your opinion on 3 points. Number one is that India has a very bad image for this the generation of MDR strains, probably that one mainly for the TBS. How much it is justified or not? I have a personal yeah, experience. 2 years back, my mother was in the hospital for a week. After that, she expired. But what I want to convey here is, in 7 days, the doctors changed the antibiotic. They were all like line 3, line 4 after every second day. So what I mean to say is the main misuse of these antibiotics is mainly from the educated part of the society. The doctors, then we go to, for example, for a very common cold, we go to a medical store. For a paracetamol, there a medical store person who is probably a B pharma or not, he gives you antibiotic with that. I think that contributes a lot. And why there are not legislations or something like that where this will be made a sort of very difficult to get an antibiotic without a prescription. My last point is regarding like as you discussed this pre-antibiotic world and post-antibiotic world probably. I want to know one of the strategies to contain infections before this pre-antibiotic world was using, particularly in this eastern part of the world, Russian part, was this use of the specific phages to kill particular bacteria. That was talked up again when there is now the lack of new antibiotics coming. What is the role or what is the status of the ICMR regarding this phage therapy?
Mm. Okay, so there are many questions in your query. So first is this is not a problem in India. We can't say that India is not progressing or India is having some legislative problem. Every country and has this problem because of one factor is also the ease in the travel. So we are the carrier and we are transferring the resistant bacteria from one country to another. So it's not a problem of India only, and India is also struggling and facing this problem and trying to contain this problem through various ways. Whatever WHO is suggesting, because WHO is the one which is playing an important role at a global level. So, for example, in 2015, WHO asked many countries to have a global action plan. India immediately adopted a global action plan and working on it. And they're also spreading this message, whatever you said, like antibiotics should be given through prescription and should not be given casually and should not be given in minor conditions, should not be given in viral diseases. Uh, in every most of the time, patients are taking medicines which is called a self-medication without asking from the or going to the doctor. They are going to the pharmacist shop and asking for antibiotic, or even pharmacists are also dispensing and prescribing antibiotics. You are right that it needs to have a strict legislation and a strict uh, um, check to all these pharmacist shop, and we need to have proper data of the use of antibiotics. They should be asked where they have been given. So that's why the surveillance team is responsible for the surveillance team also to look where antibiotics are being given. Last is what you seen, what you have said that before the pre-antibiotic era, we had some crude form of antibiotics, or like for example, curd was used as antibiotic. Heavy metals like mercury was used as antibiotic or antimicrobial. Some dyes were used as antimicrobial. If you remember, Salvarsan, the first dye which was used by Paul Ehrlich, the father of chemotherapy, that was not antibiotic, that was the dye, and dye actually help in the containment of bacteria. So there were some bacteriostatic substances. They were in the form of, as I said, curd or dye or some other heavy metals. They were given, uh, which help in the detoxification of the bacteria. So, but that was the true form. Yes, yes.
The
I
question
hope
answer
I
session
answered
is
all your
over.
questions.
Now let
So regarding the status ICMR strategy on
Yes,
phage therapy.
ICMR is also see, ICMR is a body which is the national body, and giving guidelines and giving funds for research in the innovations of the antibiotics, and they are publishing guidelines also. So they have published lots of materials and documents related to antibiotic usage, and like for example, guidelines for tuberculosis in coordination with NACO, National AIDS Control, National sorry, tuberculosis program, and similarly with the National Malaria Control Program, with the NACO, National AIDS Control Organization for HIV/AIDS program. So in most of these, in most of the national health programs, ICMR is playing a role and publishing guidelines in coordination with the agencies which is working on certain infectious diseases.
uh, thank you very much
Thank you, sir.
for the part of all the participants for posing your questions and queries, and thank you very much, Professor Ziaur Rahman Sir, for answering all the questions in a very beautiful way. So I now take this privilege to thank Professor Ziaur Rahman for this very elaborate and comprehensive discussion on science of antibiotic resistance and one health. So Professor Ziaur Rahman Saab has really discussed this topic in length and discussed all the basic and applied aspects of this antibiotic resistance, which is much needed in the contemporary times. So Professor Ziaur Rahman Saab has definitely manifested or demonstrated that when it is said that an hour spent in the company of a scholar is worth reading 100 books, Professor Saab, you have really demonstrated that spending 1 hour with you is worth reading more than 100 books. You have beautifully discussed all the basic and applied aspects of this antibiotic resistance, how it has emerged, and what are the way out, just said how we can go ahead, how we can control and prevent this antibiotic resistance, what are the global bodies and what are the gap programs that are in force, and also you have discussed various mechanisms how we can get eradicate this antibiotic resistance, and also you have discussed antibiotic resistance and how antibiotic resistance develops. It was really a thought-provoking lecture from your side, sir. So we really enlightened with your knowledge and presence. Thank you very much, sir. So with you have really enlightened us about the various aspects of this antibiotic resistance. Now, on behalf of HRDC University of Kashmir, and on behalf of Director HRDC and Coordinator HRDC, and on behalf of the participants and on my own behalf, I thank you very much for discussing this very interesting and important topic on antibiotic resistance. Thank you very much, sir. Thank you very much.
Thank you very much, Dr. Bilal Ahmed Mir, for coordinating this session, and Dr. Ishaq Mir, my good friend, inviting me to share my views on this antimicrobial resistance, as well as I'm thankful to the participants as well as the coordinator of the HRDC Center. And participants, without participation, active participation, most of the time lectures become boring. So I also enjoyed this lecture with all the participants. Thank you so much, all the participants, and with Dr. Bilal Ahmed. Thank you
Thank you
so
very much.
much.
So
Okay.
you can now close the
Okay.
share presentation, and now we may have a participant presentation from Dr. Nusrat Aziz. Dr. Nusrat Aziz, can you please share your presentation and start your participation presentation, please? Dr. Nusrat Aziz,
Yes, sir.
you can now you have a participant presentation now.
Yes, sir.
Yeah, you can share the screen now. You can start your participant presentation.
Sir, I'm doing to share my screen.
Okay. Yes, yes.
Sir, is my screen visible there?
Yeah, yeah, it is visible. You can, yes, yes, electromagnetic waves and the electromagnetic spectrum.
Thank you so much, sir. Thank you so much. I'm audible to everyone?
Yeah, 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 you are audible. You can go ahead.
Uh, a very good afternoon and assalamu alaikum to everybody. Today, I have selected the topic electromagnetic waves and the electromagnetic spectrum. As everybody is aware about this, and we are coming in contact every time with the radiations that I have selected here, that are the electromagnetic waves here. And this electromagnetic waves, this is having a broad spectrum that we all have heard about, or we all are knowing these. We have come across every time we come across these radiations. So I have selected this topic, and I have a brief presentation on this. Electromagnetic radiations. Everybody among us maybe most probably aware about the electromagnetic waves. Electromagnetic waves that
मेरे
are
उसमें थोड़ा
created
साइड
as a result
स्पेक्ट्रम
of
रखा है वो का
vibrations between an
थोड़ा
electric field and a magnetic field. In other words, we can say that electromagnetic waves are composed of oscillating magnetic and electric fields. Electromagnetic waves are formed when electric field comes in contact with the magnetic field. They are hence known as electromagnetic waves. The electromagnetic field and magnetic field, electric field and the magnetic field of an electromagnetic wave, they are perpendicular, or we can say they are at right angles to each other, and hence they are also perpendicular to the direction of propagation of the electromagnetic wave. Electromagnetic wave travel with a constant velocity, that we are knowing that with the velocity that is 3 into 10 raised power 8 meter per second in vacuum, they are deflected neither by the electric field nor by the magnetic field. However, they are capable of showing interference or diffraction. An electromagnetic wave that can travel through anything, but be that may be air, a solid material, or a vacuum. It does not require, it does not need any medium to propagate or travel from one place to another place. So electromagnetic waves, they are these are the waves, transverse waves, and without a medium they can travel. They can travel through empty space or through the vacuum. So mechanical, we can say the mechanical waves like sound waves or water waves, on the other hand, we need a medium to travel. But for electromagnetic waves, they do not need any medium for their propagation. This means that these waves, they can pass through, they can propagate through the vacuum, empty space. So these are the transverse wave, and they are making the crusts and the troughs there. Um these are transverse in nature. This means they are measured by their amplitude, or we can say height, and the wavelength, distance between the highest lowest point of two consecutive waves. The highest point of a wave is known as the this crust, whereas the lowest point is known as the trough. Electromagnetic wave can be split into a range of frequencies. We can say range of frequencies, it can be splitted, and this is known as the electromagnetic spectrum. For example, examples of these electromagnetic waves, the spectrum in which this is broadly this differentiated, or it has been expanded in the spectrum, that the examples there are the how many different radiations or the different ranges are there. We examples are the radio waves, for example. Microwaves are infrared rays, x-rays, gamma rays, etc. There are the electric and magnetic field. In this say that the electric field and magnetic field, they are perpendicular to each other. In this slide I can show this is the consecutive troughs or crusts or the troughs where we can measure the wavelength of this wave, and they are always changing they are perpendicular to each other, changing their directions, and they are always perpendicular to the direction of the propagation. When an electric field changes, so does the magnetic field. The changing magnetic field causes the electric field to change. When one vibrates, so does the other, and result is our electromagnetic wave. Electromagnetic wave travel very fast, around the 3 lakh kilometers per second, the speed of the light. You can see that at this speed, they can go around the world 8 times in 1 second. These electromagnetic waves, they have been so named that it has electric and magnetic field, and that simultaneously oscillate in planes mutually perpendicular to each other and to the direction of the propagation through the space. Electromagnetic radiations has the dual nature. That is, it exhibits wave properties and particulate properties, the photon properties or the particle properties. This is having the dual character. There are only two ways to transfer energy from one place to another place. Number first is through a particle, and another is through the wave. So these electromagnetic radiations has the property of wave, but also can be thought of as a stream of particles. Light, for example, as a light, we can say here this light as a wave. Light behaves as a transverse wave which can filter using polarized lenses. Light as a particle, if we take it as a photon, when directed at a substance, light can knock electron off a substance. That is the photoelectric effect, by which we can have the photoelectrons, and that execute the photocurrent, and we are having the effect that is known as the photoelectric effect. This is the broad spectrum here. That is the name for the range of electromagnetic waves when placed in order of increasing frequency. We are having here the radio waves, infrared rays, ultraviolet rays, gamma rays, and in between these radio and infrared rays, we are having the microwaves. In between these microwaves, we are having the visible light. These are the different. This is the spectrum where we are having, where at some places we are having the longer wavelength. We have the longer wavelength, shorter frequencies here, shorter wavelength, the longer frequencies, and we can notice here the wavelengths, longer radio waves, and get shorter up to the gamma radiations. The spectrum here is shown here, where from we can give an example how the size we can measure when we are saying the radio waves. We can give an example such like a building, or the humans like a honeybee, pinhead, so up to the atomic nuclei. So this is the spectrum here. This is the electromagnetic spectrum, which has here, I have shown here that the range in the wavelength, in the frequency, how we can measure here, and where from we can measure a portion of radiation, that is the radio waves, these microwaves, or another or other spectrum that is including in the spectrum. So first we are having here the radio waves. These radio waves are a type of electromagnetic radiations with wavelength in the electromagnetic spectrum longer than infrared light. Radio waves have frequencies as high as 300 gigahertz to as low as 30 hertz. Like all other electromagnetic waves, these radio waves travel at the speed of light in vacuum. Radio waves are these are undergoing acceleration such as time varying electric currents. So there are naturally occurring radio waves which are emitted by lightning and astronomical objects. These radio waves are generated artificially by transmitters and received by these radio receivers using these antennas. Uh, Radio waves are these are a type of electromagnetic radiation with wavelength in the electromagnetic spectrum longer than the infrared light. Radio waves have frequencies as high as 300 gigahertz, as in this spectrum shown here, to as low as 30 hertz. At 300 gigahertz, the corresponding wavelength is 1 millimeter shorter than a grain of rice. At 30 hertz, the corresponding wavelength is 10,000 kilometers longer than the radius of the Earth. Like, so this is the global this positioning system measuring the time it takes a radio wave to travel from several satellites to the receiver, determining the distance to each satellite. Radio wave pick up radio waves through an antenna and converts it to sound waves. Each radio station in an area broadcasts at a different frequencies. So the MRI, magnetic imaging, use shorter wave radio waves with a magnet to create an image. Another is the spectrum. In the spectrum, the radiations, in the spectrum, we are having the waves, that is the microwaves. They have the shortest wavelengths and the highest frequency of the radio waves. These microwaves are a type of electromagnetic radiations, as are radio waves, ultraviolet radiations, X-rays, and gamma rays. Microwaves have a range of applications, including these communication, um, radar, and perhaps best known by most people, that is the cooking. Microwave are a form of electromagnetic radiations with the wavelength ranging from about 1 meter to 1 millimeter, with frequency between 300 megahertz and 300 gigahertz. Microwave, oven commonly known as microwave, is an electric electric oven that heats and cooks food by exposing it to electromagnetic radiation. These waves transfer energy to water in the food, causing them to vibrate, which in turn transfer energy in the form of heat to the food. And these are the microwave oven, commonly here known as these electric ovens, that heats and cooks food by exposing to an electromagnetic radiation in a microwave frequency range. This induces polar molecules in the food to rotate and produce thermal energy in a process known as dielectric heating. Microwaves are used mostly for point-to-point communication system to carry all types of information, including voice, data, video in both analog and digital format. Radar, this is the radio detection and ranging, used to find the speed of an object by sending out radio waves and measuring the time it takes them to return. Then the another portion here of this spectrum is the infrared rays. These are the shorter wavelength and higher frequency than microwaves. These infrared rays, or we can say the infrared light, are a part of the electromagnetic spectrum. People encounter infrared waves every day. The human eye cannot see it, but human they come to detect it as heat. Infrared rays are these are the short wavelength and higher frequency than microwaves. A remote control that we use daily, everyone who's using in their at their own, uses light waves just beyond the visible spectrum of light, that is the infrared waves, to change channels on our TV. This region of the spectrum is divided into near, mid, and far infrared. We can sense some infrared energy as heat. Some objects are so hot, they also emit visible light, such as fire, as fire does. Other objects such as human are not as hot and not as hot and only emit infrared waves. Our eyes cannot see these infrared waves, but instruments that can sense infrared energy, such as night vision goggles or infrared cameras, they allow us to see the infrared waves emitting from warm objects such as humans and these animals. We can feel the longest one as the warmth on our skin. Warm objects give off more heat energy than cool objects. So this is the thermogram through which we can see, this is showing here a picture, that region of the different temperatures. When we put our hand on the thermogram, that shows the different temperature in the body. Temperatures are calculated by the amount of infrared radiations given off. So therefore, people give off infrared rays. Here, heat lamp give off infrared rays. So these are the regions where from we can see that how much amount of heat we are giving in the form of the infrared radiations. Then we come to the another portion of this spectrum, that is a visible light. And this visible light is the shorter wavelength and higher frequency than infrared. Electromagnetic waves, we can see here the longest wavelength, that is the red light, shortest wavelength, that is violet. This visible spectrum is the portion of the electromagnetic spectrum that is visible to human eye. Electromagnetic radiation in this range of wavelength is called the visible light, or simply we say the light. A typical human eye will respond to wavelength from about 380 to 750 nanometers. In terms of frequency, this corresponds to a band in the vicinity of 400 to 790 terahertz. Visible wavelength pass largely unattenuated through the Earth's atmosphere via the optical window region of the electromagnetic spectrum. As the full spectrum of visible light travel through a prism, uh, the wavelength separates into the colors of the rainbow because each color is a different is a different wavelength. Violet has the shortest wavelength at around 380 nanometers, and red has the longest wavelength at around 700 to 790 nanometers. Our sun produces more yellow light uh, than any other color because its surface temperature is 5,500 degrees Celsius. If the sun's surface temperature were cooler, say 3,000 degrees Celsius, it would look like reddish, like a star. Betelgeuse, we say. If this temperature, the surface temperature of the sun, this were hotter, say about 12,000 degrees Celsius, it would look like star, blue, it will look like blue. And this is comprised with the star, that is the regal. So when light enters a new medium, it bends, refracts. Each wavelength bends a different amount, allowing this white light to separate into its various colors. That's Vibgyor. So Sir Isaac Newton, that has done an experiment in 1665. He showed that a prism bends light, visible light, and that each color refracts at a slightly different angle depending on the wavelength of the color. Then we come to the another portion of this range of this spectrum, that is the ultraviolet rays. And these rays, they are shorter wavelength and higher frequency than visible light and carry more energy than visible light. So these ultraviolet rays, that is a form of electromagnetic radiation with wavelength from 10 to 400 nanometers or 750 terahertz, shorter than that of the visible light but longer than that of the X-rays. These ultraviolet rays is present in sunlight and constitutes about 10% of the total electromagnetic radiation output from sun. It is also produced by electric arcs and specialized lights such as mercury vapor lamp and the black lights. There are the various effects of these, or we can say the uses of these ultraviolet rays. The chemical and the biological effects of these ultraviolet radiations are greater than simple heating effects, and many practical applications of ultraviolet radiation derive from its interaction with organic molecules. These the radiations, it can also damage skin or give a painful sunburn, or we can use these shorter wavelength ultraviolet, these radiations, they can damage DNA and sterilize surfaces with which it comes into contact. So it kills the bacteria. Sterilization of the equipments we can use. Too much can cause skin cancers. Too much exposure of these radiations can cause skin cancer. You use can use for that, we can use the sunblocks to protect against ultraviolet radiations, or they cause the skin. These are the useful ways through we can use the ultraviolet rays, cause skin to produce vitamin D, which are good for our teeth and bones. The another part of this spectrum is these are the X-rays. These are shorter wavelength and higher frequency than ultraviolet rays, carry a greater amount of energy, can penetrate most of the matter, and they are having a very shorter wavelength and the higher frequency. As we are going to the shorter and shorter wavelength and the higher and higher frequency, they are having the highest penetration. They are having the
Dr.
highest
Nusrat, can you
penetrating
please
power.
conclude now? Dr. Nusrat, can you please conclude now? Because we have
Okay,
so
sir.
many other
Okay.
presentations.
So this is the X-ray that we can use. We are having the various uses of the X-rays. It can show the image, the light part of an X-ray image, indicating a place where the X-ray was absorbed. So too much exposure can cause cancer, lead us to dentist protest against from unnecessary exposure. So we can use in the these are having the uses in the engineering to check for any cracks in the structures. Another portion, the last portion, is the gamma rays. The shorter wavelength and the higher frequency than X-rays carry a greatest amount of energy and penetrate the most. So use of radiation treatment to kill the cancer cells can be very harmful if not correctly used. So these exploding nuclear weapons emit these gamma radiations. So these are the brief summary of these electromagnetic radiations. They can travel with the speed of the light in vacuum. They have the different wavelengths at different frequencies. Where we are having the longer wavelength, there will be the lowest frequency, shorter wavelength, highest frequency. The higher the frequency, the higher will be the energy. So this is the spectrum which is showing the every portion of these radiations in this. Thank you, sir.
Dr. Bilal, please invite the next speaker. I think perhaps Dr. Bilal has got disconnected. Dr. Feroz Ahmed Bhatt, may please start.
No, sir. I'm connected. I'm connected, sir. Dr. Feroz can start his presentation and share the presentation with the participant. Dr. Feroz.
Okay, sir. Thank you. Thank you, Dr. Bilal, for allowing me to present my topic. Respected Professor Geer Sahab, dear participants, a very good afternoon and assalamu alaikum to all of you. As per the directions of our worthy director, worthy coordinator of this very refresher course, to select the topics which are common to all. Since I am from the mathematical field, it becomes a bit difficult for all people to understand every difficult equation of mathematics. That's why I have chosen a very simple topic, the day of the week from the date. This, in fact, tells us how to calculate or how to check which day falls on a particular date. For instance, if you select say 14th April 684, you can by applying this very mathematical formula, you can say which day will be it, whether it is Sunday, Monday, Tuesday, or any other day of the week. The equation is very simple. It involves simple mathematics, not difficult mathematics. But before introducing the equation, I will just make use of some, explain some definitions involved in the equation. So first is integers. This is common to all. The numbers 0, 1, 2, 3, 4, so on, and -1, -2, -3, 4, -4, so on. These we call as integers. Integer. In fact, these are actually the counting numbers, the numbers which you use in counting the objects. Uh, when you count the pages of a book, you say just 1, 2, 3. So these numbers and their negatives, including 0, in aggregation, we call these as integers. So these numbers will be called the integers. We also have the numbers which are not integers, non-integers, like 3.5, 3.56, 13 by 5, and so on, which you do not use in counting. So this is I'm going to use in the formula. Next is a kind of operation which is not the among the fundamental operations of arithmetic. We have four operations in mathematics, four fundamental operations, which is like addition, subtraction, multiplication, and division. This is a bit different from that. In ordinary sense, sometimes you say uh, equations balanced, or you say some number x is equal to y. That's in the ordinary sense. You can say 2 is equal to 2, 7 is equal to 7, but that's in the ordinary sense. But in we have modular arithmetic concept here, which I'm going to tell you in the sense of congruence. So what by a congruence we mean here, given any two integers a and b, I listed above integers, so these are two among them, any two. Given any two integers a and b and a positive integer m, we say that a is congruent to b modulo m if m divides their difference. By division, what we mean, we can say 2 divides 10, because 2 multiplied by 5 equals to 10. Everybody knows what by division we mean. So we say a and b are congruent modulo m if m divides a minus b. Here we can say a is congruent. We write this information in this way. There's a mathematical symbol for this. A is congruent to b modulo m. So this you can say in the sense of equality, the a is equal to b, but this is not in the ordinary sense. Like you say 2 equals 2, but here you can see in the example, if a is 18, b is 3, and m is 5. Suppose I choose these three numbers in particular, then you can see b minus a equals to 18 minus 3, which is 15. And 15 is divisible by 5, because 5 multiplied by 3 equals to 15. So I can say 18 is equal to 3, but it's under the modulo 5. This is not in the ordinary sense. You can never say 18 is equal to 3. But under this operation, under this congruence modulo, you can say 18 is equal to 3, but under modulo 5. Similarly, you can have different moduли. So this is the definition of congruence. I am just going to make use of two definitions here. One is the congruence, and next is the greatest integer function. This says if x is any this says if x is any number, then the greatest integer of x is defined as greatest integer less than or equal to x and is denoted by this symbol. Here I am saying x is any number. So I am coming out of integers. You can have x any number like 3.4, you can have 7.9, or you can have integer also x, any number, not only integers. For instance, what by this greatest integer function mean? This extracts the integral part of a given number. For instance, if you have 5.6, 5.6 has greatest integer equal to 5. If you go on the number line, you just have to select the greatest integer lying to the left, very close to 5.6, but from the left side. When you go on the number line, you have 1, then 2, then 3, then 4, then 5, then you have 5.6 or 5 points or whatever. All these numbers between 5 and 6, except 6, these numbers have greatest integer 5. Similarly, 4.33 is 4, minus 1.2 is minus 2. It's just what we do here, we just extract the integral part of a given number. I think it's clear to everybody. This is mathematical,
Yes.
so I need to explain these things. This is a number line on which you can select. For example, if you select the number between 3 and 4, any number lying between 3 and 4 has greatest integer 3. So here you can have 17 by 3. If you take 17 by 3, the value comes out to be 5 point something. So what would be the greatest integer? Just take out the integral part, that's 5. Similarly, minus 9 by 4 is minus 2.25, which is minus 3. So this is very simple. Now we will see how to calculate the day of the week from the date. So there will be first of all the certain rules which govern the equation actually. So I will select the date. You can select any random date. Let me suppose I have 9th November 1877. So this is a very back date. I want to see which day will correspond to this date. Now, if you select any date like this, 9th November 1877, it defines 4 integers, which we denote by N, M, C, and Y. Let us see what these 4 integers are. Let N denote the number of the day in the month, so that N is 9 for the 9th November 1877. So N denotes the number of the day in the month. In this case, N is 9. M denotes the number of month counting from March. So the origin for this M, I mean, starting value for this very M is March. These are the rules which are used in this formula. M has been fixed to be 1 for the March. I mean, we had to start from March. March is 1, April is 2, and so on. January 11, and February is 12. So these are the values. These values we have to insert in the equation. This explains the rule how to select the values of these 4 numbers, which every date defines, like N, M, C, and Y. Thus, in our case, we have actually the date, our date is here, 9th November 1877. So M actually denotes the month, number of the month in the year. We have November here. So if we go in this very table, we have M12. Sorry, we have November is here, 9. We have M equal to 9 for the date 9th November 1877. Actually, this convention arises because of the extra leap year. Because extra leap year is added, I mean the additional day is added at the end of the February for every a leap year, extra day of the leap year. I'm sorry here. So we have C. 4 numbers are defined there: N, M, C, Y. What is C? C denotes the hundreds in the year. So that C is 18. You have 1877. The date is here 1877. We have C. C denotes the hundreds, number of hundreds. It is 1800. So C is 18. And Y denotes the remaining value of the year. We have 1877. What is remaining? That is 77. So Y equals to 77. So we have 4 values, N, M, C, Y. You have seen how to select the values of these 4 values for any date. Next is this one here, D. If D denotes any date of the week, then we take these values. These are I mean, we have to use these. If the value of D comes these values, then we have to, I mean, jump upon the corresponding date. So D is 0 for Sunday. I mean, if in the equation value of D comes out to be 0, we will say the corresponding date is Sunday. If it is 1, then it is Monday, and so on. If D is 6, and Saturday. Then the value of D is actually, this is the formula. I mean, from this we have to solve this equation. We will get some value of D. Like if D is 6, then you will say it is Saturday. If D is 5, you will say it is Friday, and so on. So these are the values. this D is congruent to, the congruence I have explained it. D is congruent to n plus 2.6 m minus 0.2. Then these brackets are actually those very the notation of the greatest integer function. What will be inside the value? Suppose 2.6 into m minus 0.2, its value is suppose 7.4. Then you will apply the greatest integer, you will have 7 as a value. Then you have to solve this. There's values inside. You get finally some value. You can see here I am using greatest integer, so everything in this each term in this equation will be an integer. N is integer, 2.6 m minus 0.2 will be some integer, y is integer, y by 4 will be some fractional number. Then you are applying greatest integer, so everything will be integer. Then you have to add, then you have to see is D congruent to that. If D minus that is divisible by 7, then value of D will come out. Now we have used it here L also. You can see D I have defined it and already defined M, Y, C, all of these are defined. We define L here. L is considered to be 1 for a leap year and 0 for a non-leap year. Now the question is how to check which year is a leap year. The leap years are those which are divisible by 5. For instance, if you have 2020, that's divisible by 4. So you can say it's a leap year. But among these leap years, so the definition is here like this: the leap years are those which are divisible by 4, except the years which are ending. I'm sorry, here ending with 00, which are leap years only if divisible by 400. So leap years are actually those which are divisible by 4. But if the year is ending with 2 zeros, then the formula for the rule for checking whether it is a leap year or not is that you have to divide by 400, not only by 4. I have given some examples here. For instance, if you take 2016, 2024, these are leap years as they are divisible by 4. While if we take 1900, you can see 1900 is really divisible by 4, but still it is not a leap year. Because for such a year, if it ends with 2 zeros, then that time you have to divide by 400. You can see 400 does not divide 1900. So 1900 is not a leap year, even if it is divisible by 4. For these types of years, you have to divide by 400. Now let me come to the exact formula. Calculation for a day corresponding to 9th November 1877, which I have selected. I will calculate the corresponding day. I will check which day it is. Here I have defined above: n is 9, m is 9, c is 18, y is 77, and l is 0. Because we have 1877, it's not divisible by 4 because it ends by an odd number. So it's not a leap year. So we have all these values. Just substitute here. D equals to 9 plus 2.6 into 9, and all these values you can put here. Then you will have these values. D equal to 9 plus 2.3. Okay. Here I have 9 in the last step on the screen. D is congruent to 9 plus 23.2. It's the greatest integer. You have to extract integral part. That's 23 plus 77. Then you have 19.25. It will be 19. Similarly, 4.5 will be 4. Then you have 36 minus you have 0.81. It is 0.81. 0.81 is not an integer, but to the left of 0.81, what is the greatest integer? That is 0. That's why I'm selecting 0 here. So the value of D finally, after adding all these and taking the, subtracting that adding, you get finally D congruent to 96 modulo 7. But you can see from the definition of congruence, 96 is congruent to 5 modulo 7. Because if you subtract 5 from 96, you have 91. You can divide by 7. So you have 96 is actually congruent to 5 modulo 7. So you can say D is equal to 5. D is equal to 5. But you can see when D is 5, what is the value? What is the date corresponding to this day? When D is 5, it's Friday. So I can say 9th November 1877 corresponds to Friday. Corresponds to Friday. So required day is Friday. This day is actually the date of birth of Dr. Allama Iqbal, rahmatullahi alayh. The
Farah sir,
formula holds. The
Farah sir,
formula holds for
can you
any
conclude
date.
now? It is already too late.
Bus, I have
Sorry,
completed.
sorry,
Bus,
sorry to
this is the
interrupt.
last, sir. Next is thank you. You can see
Okay,
here.
thank you.
You can see. The formula holds for any date after 1582, following the adoption of Gregorian calendar at that time. Thank you. I can tell you one more thing here. This formula is in fact applicable to any future date. You can select any date in future and you can calculate the corresponding day. Thank you for hearing me.
Thank you very much for your informative and interesting talk, Dr. Feroz Saab. And I think we are already very late, and we should conclude this session right now with this presentation. And the rest of the two presentations, Ifat Bashir and Dr. Kausar, will present in the afternoon session. Thank you very much.
Thank you, sir.
Please be
Yes,
back
Saab,
at,
you want to say anything?
yes, all participants are requested to join back 2 o'clock on time. Please be on time. And either Raja Nisar Ali or Wasim Rashid, who is going to moderate the afternoon session? Raja Nisar Ali, are you there? Can you hear me?
Yes, sir. Yes, sir. I can hear you.
So are you going to moderate the afternoon session?
Actually, Wasim had dropped a message in this group.
Where is he? Where is
available? I'm ready.
you are ready? Dr. Wasim Rashid, are you there?
So no problem, sir. I will moderate.
Dr. Wasim Rashid is not present, so you may please moderate.
Yeah, thank you, sir. Thank you.
Okay, okay, sir. Thank you very much, sir. Thank you very much, Isaac Saab, also for this wonderful program.